Good morning, everyone. Thank you, Dr. Vecina and Dr. Rodriguez, for the invitation. It's my great pleasure to be here. It's my first time in Spain, and so I'm very pleased to have the opportunity to give this presentation to you. And again, I'm very happy to answer any questions throughout the day. What I'm going to tell you is, I think, very relevant to whether you are doing research on human reproduction or whether you are in the clinic taking care of human embryos and their developmental potential. And it's about genetic instability in embryonic development. So we all know that human embryonic development is rather inefficient, and it is thought that chromosomal abnormalities are a leading cause of that inefficiency. And among those causes are two, principally, which are abnormality in meiosis. These are well known and quite well understood. And then also abnormalities in mitosis, which are somewhat less well understood, both in terms of how they arise and what the consequences are. But we all know that only about 1 in 4 fertilized eggs result in a pregnancy, and perhaps sometimes even fewer than that. So human reproduction is not very efficient, and it is thought that these abnormalities play an important role. So my talk is going to focus on genetic instability during preimplantation development, and specifically during mitosis. So the questions are, what are the developmental consequences of mitotic segregation errors, and what are the clinical consequences? So what does this mean for you? Chromosomal abnormalities can be caused by abnormal chromosome spindle interactions. And this is from a review from Terry Hasselt and Patricia Hunt, and it shows that you can have abnormal chromosome spindle attachment. The result, this would be normal, so you have replicated sister chromatids, and in a normal cell division, they segregate to opposite poles. If you have abnormal chromosome spindle attachments, they can either both go to the same pole, or called non-disjunction, or what also seems to happen quite frequently is that there is premature segregation of sister chromatids in meiosis, so-called meiotic early premature segregation. That can occur in M1, but also in M2. So there's good evidence for meiotic segregation errors as a leading cause of miscarriage and congenital abnormalities. There is somewhat less of a clarity on the significance of mosaicism, and there is an uncertainty on what to do with mosaic embryos. And so just very recently, there was a practice committee of ASRM that talked about what to do with genetic testing for aneuploidy, and there were clear recommendations of what to do with meiotic aneuploidies, but there was no stronger recommendation of what to do with mosaic embryos. And this is also reflected in the very recent article in the Wall Street Journal, as IVF testing spurs a debate over mosaic embryos. And so there's a disagreement of what should be done with embryos that are called aneuploid, with some euploid cells and some aneuploid cells due to mosaicism. So there's a disagreement. And so I think it is important to better understand how it arises and what the mechanism is and what the developmental consequences are. So mosaicism, of course, means that the defect arises during mitosis, because mitosis means not all cells will have the same chromosomal abnormality. And so this is an example, and it's actually done on single blastomeres at the cleavage stage. You see here this chromosome gain, here again a chromosome gain, here a reciprocal chromosome loss, and here a reciprocal chromosome loss. So you have clear mitotic aneuploidy. In a meiotic aneuploidy, you would either have a uniform gain or a uniform loss. But here within the same embryo, you have both loss and gain, which means it's mitotic, and so it's mosaic. So having these reciprocal outcomes clearly confirms that this is mosaicism, because it is different in different cells. There's the gain and the loss here, so this is a very reliable call of mosaicism. So what did we learn about the mechanism of mitotic aneuploidies? And here I will show you a series of experiments that were done in the field of stem cell research and reprogramming. And they led us to understand not only how mitotic aneuploidies occur in that context, but also how they occur in human fertilized embryos. So what I'm going to show you, you can see that as an experimental model system for the mitotic aneuploidies in human preimplantation development. So, a brief introduction. One goal, or a so-called holy grail of pluripotent stem cell research and regenerative medicine is to make stem cells that are matched to a patient. So a patient, for example, with diabetes or another degenerative disorder such as Parkinson's can donate a somatic cell, and that cell is made into a stem cell and then made into the cell type that's affected, so it can again be transplanted into the affected patient to establish a cure. And of course, this concept could lead to cures for multiple disorders, including diabetes, Parkinson's, spinal cord injuries. That's the hope, and there are many ongoing groups and research programs that have this as the goal. And so what we did, we used a sort of unique approach, but this was actually the original approach, how people envisioned doing that, is to do cloning or somatic cell nuclear transfer to generate these pluripotent stem cells. And you can see here that it uses a human egg and to make that stem cell and then for differentiation. I'm going to show you briefly how that works. Many of you might already be familiar, but for those who are not, this is a quick summary. This is the egg, the unfertilized human egg. The genome of the egg is removed and replaced with a somatic cell of the patient. Instead of fertilization, an artificial activation pulse is used to allow developmental progression. And so this is how it's done. That actually shows the process of enucleation of the egg. A little hole in the zona is made. In this case, that's an animal egg. This is not a human egg, this video. As some of you might easily recognize, human egg looks slightly different, but in terms of size, it would be quite similar. Here you see the chromosomes over here. So that egg is enucleated. And then a somatic cell is either injected or fused to that egg. So here comes the somatic cell. In the pipette, again, a piece of the zona is removed, and it is then injected. In human eggs, I usually do cell fusion. It works better, but in this case, this shows the transfer of the somatic cell via injection. And so we succeeded, after several years of optimizing that process for human eggs, we succeeded in making blastocysts, shown here. So that's a human blastocyst of the somatic cell nuclear transfer. This is a blastocyst with the inner cell mass. These are the stem cells that were derived from them. And as you can see, what was originally a skin cell now can make many different types of tissues, including neurons. And we showed, this was from a type 1 diabetes patient, that you could make beta cells. All the green clusters up here, these are hundreds of cells, each cluster. All the green are insulin-producing cells. They're marked here with an insulin GFP. This is absolutely spectacular. You can make relatively high purity beta cells from these reprogrammed stem cells. And you see these are very uniform. And of course, we and others are hoping to use that for transplantation into patients with diabetes. What this shows here is that these cells, they can restore glucose homeostasis in mice. This is a mouse without the graft, with high glucose. These are mice with the graft, and that maintain normal glucose homeostasis. So this is not the topic of this talk, But it shows that this can work. We can get human somatic cell nuclear transfer to work in humans. We can get them to develop, and we can make those stem cells. However, quite frequently, the embryos that are made after somatic cell nuclear transfer arrest at the cleavage stage. And so that was an obstacle to the generation of these stem cells. And we asked the question, why is that? And what we found is that these blastomeres often contained karyotypic abnormalities. Here again, a chromosome array shows gain and loss of chromosomes. That was done with Nathan Treff of Genomic Prediction, who actually tomorrow will talk about what to do with mosaic embryos. So a very related topic and relevant to what I'm going to talk to you about today. So in order to understand how these chromosomal abnormalities arise so frequently, we designed some experiments that asked, well, is this a problem of spindle chromosome attachment of the sorts that I showed you earlier in the schematic, where the spindle attaches to only one sister chromatids or not the other? Or is this something else? And so the way we did that, we transferred a replicated somatic cell. So that has a replicated sister chromatids and asked, can it segregate those chromosomes normally at the second meiosis? So this is right after transfer. And of course, that involves spindle chromosome interactions. And we asked, can it extrude that second polar body normally? But then again, you can ask at the first mitosis, can it segregate those chromosomes into two daughter cells? And if there is a difference between those two, you could point to a mechanism. So, in meiosis, it turned out that if you transfer those replicated chromosomes into the egg, the human egg, then you can see that these chromosomes segregate normally. One group, the other group. Normal segregation, polar body extrusion, is a pseudopolar body, and the pronucleus forms. So there was no evidence that there is a problem of spindle chromosome attachment that leads to segregation errors after somatic cell nuclear transfer. Now, one cell cycle later, at the first mitosis, it looks very different. What you see is this sort of bridge, but you also see fragments, so bridges and chromosome fragments. Again, these two errors are not typical for a chromosome spindle attachment error where whole chromatids are missegregated. Rather, they indicate that there is an entanglement, perhaps, or a break in the chromosome. Now we'll get back to that. So, at the second meiosis, we found that in the chromosomal microarray that we did on these pseudopolar bodies, the DNA content was normal, so the segregation was normal. But at that first mitosis, we saw those aneuploidies form. Actually, this is later mitotic division. This is from a blastomere. But this is at the first human mitosis. So here you look at the first cell division of the human egg. Then, in these blastomeres, we saw evidence of DNA damage. This is gamma H2AX. That's stains, double-strand breaks, both in mitosis and in interphase. We saw those foci of gamma H2AX. RPA stains single-stranded DNA. That shows again, this is DNA damage. It's not a problem of spindle chromosome attachment. And then these micronuclei, which is also what you commonly see in IVF human embryos. Just that the frequency by which this did occur after somatic cell nuclear transfer was very, very high, much higher than after IVF. To better understand what was going on and the mechanism, we resorted to mouse cells. You can appreciate that it's difficult to do experiments in human eggs at these larger numbers. although we are very fortunate to have one of the few oocyte donation programs for research in my lab. But mouse cells are often very useful to dissect the molecular mechanism. So what you see here in the mouse, we found that it was very similar. At the second meiosis, chromosomes could be normally segregated, but at the first mitosis, we found evidence for bridges, DNA damage, and lagging chromosomes. So again, what we saw in human pre-implantation development, we could also see in the mice after somatic cell nuclear transfer. So the mouse was a very suitable model system to study what was going on. And surprisingly, we found that it depended much on what kind of nucleus we transferred into those eggs. So this shows the frequency of segregation errors. And in eggs that are untreated, you would expect it to be low or less than 5%. But when you transferred either embryonic stem cell nuclei or somatic cell nuclei, you saw a rise of these errors in 20 to 30% of the eggs. So that's fairly high, and certainly much higher than the background levels. So which pointed that epigenetic features of these somatic cells were responsible for these segregation errors. We then asked, well, does it have anything to do with gene expression? Because a somatic cell, of course, has a different gene expression pattern than an egg, and so one thing that changes will be gene expression. But as you might know, the early embryo does not have gene expression. It is transcriptionally silent. And indeed, when we added to the transcriptional inhibitor alpha-mannitine, there was no change in the segregation errors. So this was not related to the change of gene expression after nuclear transfer. In the mouse, most of the zygotic gene expression starts at the 2-cell stage. In human, it starts about between the 4 and 8-cell stage. And that's, so the problems in the chromosome segregation was before there was any gene expression. It was at the first cell cycle. So this pointed out that the problems, the defects we saw, were not due to problems in gene expression. They were related to something else. So what could it be? Well, so far, let me summarize the conclusions. The chromosome segregation errors were not a spindle defect. They're associated with DNA damage, and they're due to cell type specific difference unrelated to gene expression. They occur only after the first interphase. The second meiosis was normal. So this was somewhat of a surprise because we often see chromosome segregation errors as these spindle attachment errors. But what else could cause these errors? Could it be DNA replication? For normal segregation of chromosomes, of course, you need a normal duplication. You cannot segregate chromosomes that are not fully duplicated. So that was an obvious, it wasn't quite obvious, but it was something that we certainly had to think about. And the types of errors that we saw were very similar to the ones that you see in cancer cells. And it is known from studies in cancer cells that problems such as bridges or chromosome fragments that lack a centromere are not due to spindle attachment errors. This would be, spindle attachment errors would be a mitotic defect, but these errors arise pre-mitotically during DNA replication, bridge and chromosome fragments. While if you have a centromere, this can arise through mitotic spindle chromosome attachment errors. But this was mostly what we saw. So in our experiments, we found that the errors that we saw carried the hallmarks of DNA replication-dependent errors. Also, we looked at DNA damage, and again, as I showed you, gamma H2X, which marks double-strand breaks, and RPA, which marks single-stranded DNA, these are overlapping, and what we found is that they arouse predominantly at the end of S phase, at G2. So that's nuclear transfer in G2. You see this rise of DNA damage, both in the first cell cycle and the second cell cycle. So when exactly is S phase? We commonly think of the cell cycle as G1, S phase, and then S phase ends, and there is G2, where the cell waits for cell division. M phase, where it undergoes the division. Then again, it starts G1, S, G2. So this is a time scale for the mouse. The human time scale is extended a little bit. You would have S phase between maybe 6 to 14 hours, and then M phase more around, M phase entry more around 22 to 25 hours. But the principle is the same. In mouse, S phase begins only about 3 hours after fertilization and ends 8 hours. And you can look at this by EDU staining or BRDU staining. So we think of G2 is that S phase is done. And in fact, if you try to stain for incorporation of EDU or BRDU, you wouldn't see very much. But so I ask, well, is DNA synthesis still ongoing? Excuse me. And you can also answer that by addition of acetylcholine, which is an inhibitor of DNA polymerase. When you add acetylcholine at 12.5 hours, so well after S phase, you found an enormous increase in segregation errors at that first mitosis. Also, at 13 hours, 13.5 hours, and 14 hours, you still find it. Only when you add acetylcholine here at pronuclear envelope breakdown, as cells enter mitosis, you don't find an increase. That is 0 in the mouse. It's a very reliable segregation. The mouse has many fewer errors than human preimplantation development, and therefore is not the best model to study mosaicism. But you can see that S phase really only ends here. This is called G2, but we shouldn't think of it as, we should think of it more as part of S phase. This is when the cell cleans up the ends of S phase, the open ends, when it finishes those areas in chromosomes that have not been fully replicated. M phase entry only occurs when the DNA is fully replicated, but sometimes it may not be fully replicated because the S phase really extends until right here. So, okay, there is the second cell cycle. Again, it's the same thing. You can see here S phase ends when you do EDU staining about 17 hours, but the DNA damage arises really in G2. But G2 does involve S phase, as we now know, and so most likely these are areas of DNA synthesis or late DNA synthesis that has not yet occurred. And it is probably these areas that result in chromosome segregation errors at mitosis, because those are the ones that are increased after nuclear transfer. So what have we learned from these studies? Genetic instability during mitosis is caused by abnormalities in DNA replication. Mitotic abnormalities are associated with DNA damage at the end of S phase in G2, which I think of as still being S phase. DNA damage causes checkpoint activation, resulting in cell cycle arrest. This is, we know from a large body of literature, that DNA damage is a very effective way to stop the cell cycle. There is a multitude of proteins that detects DNA damage and thereby delays it. So, how is that relevant to mitotic to mosaicism and human preimplantation embryos? What I showed you were experiments to dissect the mechanism with a model system, but how does this relate to what many of you do in the lab? Human preimplantation embryos also carry the hallmarks of replication-dependent segregation errors. For example, they're a bit more difficult to detect because the cells are asynchronous. We cannot as readily study them, arrest them, or pick them at specific time points of the cell cycle. But you can see here this bridge is reminiscent of a bridge that is DNA replication dependent. Here you see a mitotic figure with these chromosome fragments. It lacks a centromere. So again, these are the two hallmarks of replication-dependent errors in human preimplantation embryos. Together with Nathan Treff, we found that micronucleation, so mitotic errors, correlates with micronucleation. Micronucleation is not something you see easily through bright field microscopy. You really have to do DNA staining to detect it. You can see here this little nucleus on the side of the main nucleus, and that correlates with the chromosome gain, while sister blastomeres that do not have the micronucleus have a corresponding chromosome loss. So these can be segmental. You can see here the reciprocal segmental loss and gain, and here as well. These mitotic errors can occur in a segmental manner and therefore involve the breakage of the DNA at this position. And that's also seen in micronuclei when you perform staining. Again, for example, this micronucleus here does not have a centromere, while this one here does. So that directly points to a broken chromosome here in cleavage stage embryos and here in the blastocyst. There is chromosome breakage going on during these mitotic divisions. And also, in IVF embryos, you find extensive evidence for DNA damage, replication-dependent DNA damage, RPA marking single-stranded DNA, and gamma H2AX marking double-strand breaks. So, we know that from several studies, nowadays people do mostly chromosome and nucleus screening at the blastocyst stage, and their mosaicism is not quite as high as at the cleavage stage. It decreases. And that looks like a correction. But what it really is most likely it is an arrest of cells that are abnormal. And you can see here that arrested cells that do not progress from the cleavage stage, they have tremendous amount of damage in their DNA, often multinucleation, and they progress from, so that means many micronucleated DNA fragments, and these are arrested. At the cleavage stage, you find micronucleation, but then these progress to these arrested ones that have a lot of more micronucleation and do not divide. They have lost the ability to divide. These are, for the most part, excluded blastomeres that do not contribute to further development. At the blastocyst stage, you find a reduction of these, likely due to selection and elimination of these cells. So the conclusion from this is that the human IVF embryos show the hallmark of replication-dependent DNA damage. This damage is likely relevant to reproductive potential in humans. Why is this relevant? Well, the DNA replication is actually the first thing the embryo does. The first thing the embryo needs to do is to duplicate its DNA. It does that before it expresses any genes. Before it does any transcription, it has to duplicate its DNA. It's the first act of growth in order to divide. And that's also already where it first fails. DNA replication checkpoints affect cell proliferation. The cell cycle arrest will result in the exclusion of cells. And so even though there is some tolerance to a reduction of cells, there is a limit in what can be tolerated. And that is well exemplified by the lack of an inner cell mass in blastocyst stage, which is commonly seen. At the morula stage, in order to specify an inner cell mass, you need inner and outer cells. If you do not have a sufficient number of cells, there will be no inner cells, meaning an inner cell mass can't be specified. So the number of cells that is formed within a limited time window is a key determinant of cell specification and developmental potential. In other words, the duplication of the DNA is a limiting factor in whether the embryo can make enough cells at developmental benchmarks to specify specific cell types. And the earliest one is the morula stage. And so we do know that at the cleavage stage, you have a high percentage, a high frequency of mitotic errors that can result in DNA damage and cell cycle arrest. And therefore, this will result in the embryos that you exclude because they have a low grade, they have a low cell number, they don't have an ICM, and that is very common. So what does this mean? What does our new knowledge of mitotic segregation error mean for you? And how does it differ from what we know about meiotic errors? If you put the two next to each other, they're very different. The chromosomes are intact in meiotic errors. There's a whole chromatid error. In mitotic errors, you have DNA damage, you have chromosome breakage, which can result in cell cycle arrest. And so that means the cell cycle compatibility is compromised. There is a negative selection of those cells built into the DNA. While meiotic errors, there is cell cycle compatibility, and you can readily find blastocysts with a meiotic gain of chromosomes. So these have implantation potential, while these abnormal cells will get eliminated for the most part naturally. Here, spontaneous correction is possible. Here, it's very unlikely because it's hard to imagine how broken chromosomes would be resealed. The mechanism here is the aging of chromosome cohesion, while here it is DNA replication dependent. But in a PGS assay, or in our minds, the two look very similar. The underlying biology and molecular mechanisms are completely different. But when we perform an assay, such as a PGS assay, it is deceivingly similar. And because it is deceivingly similar, we might think we have to take the same action. If you have an aneuploidy that is caused by mitosis, you think it's aneuploid. That makes you worried. But the biology, we shouldn't be treating those as the same thing. It is very different. Even as the assay gives you a similar signal that resembles each other. So the clinical action, I think there's good consensus that the exclusion of embryos with whole chromosome aneuploidies caused in mitosis is meaningful. But here, you have this built-in biological mechanism for quality control that is the DNA damage signaling, cell cycle arrest, and self-elimination. These cells, for the most part, will not contribute to the developing embryo because of that mechanism. And so that's why there are reports of mosaic embryos that result in normal children. So the primary cause, and here I'd like to make some, I would say, bold statements. I do think that the primary cause for developmental failure in the human species are abnormalities in the duplication of the DNA, or difficulties in duplicating the DNA. DNA duplication is a demanding process. It has to be done within a relatively short time. It's 2 times 3 billion base pairs. There are structural elements and repeats in the human genome that can obstruct the progression of DNA replication. There are unstable areas and more stable areas. It is a very challenging process. And so it is not surprising that it actually is developmentally consequential. DNA replication checkpoints and cell cycle arrest provide an effective means to arrest development of abnormal cells. And so this is actually, I think biologically, this is a very meaningful way for nature to prevent the growth of an embryo that is abnormal. By using the signals of cell cycle progression, by using DNA damage signal to arrest an embryo with suboptimal developmental potential, nature makes sure that the cost is low. Of course, if cells just kept dividing, if cells of an abnormal embryo just kept dividing, then an abnormal mass of cells could form. But because biology has built in mechanisms to induce cell cycle arrest, it prevents the biological costs of a pregnancy very early on at the cleavage stage before implantation. So I think of these issues, I think of these problems with human preimplantation development as a most effective quality control mechanism there is in human reproduction. It is the principal mechanism of natural selection occurs right there, and you see it in the dish when you look at preimplantation embryos. That's where abnormalities are removed that could result in abnormal development, but they will never make it because these abnormalities are detected at the level of abnormalities in DNA replication. These can be epigenetic problems, such as we saw after somatic cell nuclear transfer. This could be genetic problems. There could be a variety of problems that lead to issues in DNA replication. And so an important goal of the research will be to understand why are there issues with DNA replication. What exactly leads to those problems? And I think when we understand that, we will know a lot more about human preimplantation development and us as a species. So I'd like to thank the people who have contributed to this work. At Columbia University, Glorin Cha was the postdoctoral fellow with whom I've done these chromosome segregation studies. Lina Sui has developed beta cell differentiation. I showed some data from her with Brian Gonzalez. Mitsutoshi Yamada has performed many experiments with me on somatic cell nuclear transfer. And then Daniel Kort, an OBGYN fellow, has done with me the analysis of human preimplantation development. I'd also like to thank Mark Sauer and people from CWRC, Columbia University, Nathan Treff, with whom I've collaborated for many years of genomic prediction for karyotype analysis. Thank you.